Hi, my name is Madison Thompson, and my protein is acetylcholinesterase. The mutation that I'm studying is the bonding of serine to the active site of acetylcholinesterase instead of acetylcholine, and how that impacts the function of the protein. So, the normal function of this protein is enzymatic. Acetylcholinesterase is responsible for breaking down the chemical acetylcholine, which is triggered by the nervous system to react to potential toxins and regulates the body's homeostasis, especially through secretions. For example, acetylcholine triggers tears, oral secretions, and snot to come from the body, and if it's not stopped, they'll continue indefinitely until mortality because they won't know that the nerve agent is gone. So the responsibility of acetylcholinesterase is to break apart acetylcholine and stop communication between nerve and muscle cells and allow the body and the organism to return to normal function. I'm learning about this protein because I'm terrified of snakes and I want to understand how their venom works. Acetylcholinesterase is found in the synapse between nerve and muscle cells, and that is where it can easily break down signals because it's the midway between the two. And it was first found in electric fish and is found in animals, not plants. So this is my model. I used blue pipe cleaners here to show alpha helices, and I used green pipe cleaners to show beta pleated sheets. This is the active site, and it would be in the middle of the model, or the protein. So here we have the active site. And this blue, as you can see here, is an anionic site. This is where hydrophilic molecules bond to, and this is the site of hydrolysis where the molecule is broken apart. So if you have acetylcholine and it bonds here, then it bonds to both sites, and at the hydrolysis site, it's broken down by water, and here it's just bonded. So this breaks down into choline, and it breaks down into acetic acid. This is acetic acid, and then this is an ionized form of acetic acid with an extra hydrogen. However, when normal function isn't happening, so when serine, a toxin, is bonded to this molecule, it bonds to the hydrolysis site. It's hydrophobic, so it won't bond to the anionic site, and it refuses to break down. It can break down to an extent. So if a water molecule is added, it will break apart to this and this, but it's just portions of the nerve agent. And this charge here stays bonded to the hydrolysis site and just continues. Every time water happens, it just gives this off. So because this is here, acetylcholine can bond to the anionic site, but it can't bond to the site of hydrolysis, and therefore the molecule cannot be broken down, and the nerve agent will continue to cause communication between the nerves and the muscles, and that is detrimental to the body's function. So as I kind of just explained there, the protein becomes less active when serine bonds to it because there can be no hydrolysis. So going back to the chemical makeup of acetylcholine is an abundance of methyl groups. So as you can see here, we have the methyl groups here, and here at the end. So acetylcholine is hydrophilic, meaning that it is polar and water-loving. It is routinely broken down through hydrolysis to supply the body with needed release from messages between nerve and muscle cells. Acetylcholine is positively charged, so it is acidic and likely donates protons, and it has a formal positive charge of 1, so it is also polar, supporting that it is hydrophilic. Acetylcholine has a molecular weight of 146.21 moles, which is almost dead even between the smallest amino acid, which is glycine, and that has a molecular weight of 75.07 moles, and the largest amino acid, which has a molecular weight of 204.22 moles. So acetylcholine is hydrophilic and can participate with hydrolysis and forming hydrogen bonds. The main functional groups present in serine, or the mutated version of this protein, are phosphonate, and that is hydrophobic and nonpolar. So serine is nonpolar, it has a formal charge of 0, it cannot be broken down during hydrolysis. Another important quality is it is miscible with water, meaning the two can be mixed without dissolution. So even when I put these two molecules together, they're going to remain like this in the jar. Nothing's going to happen. They're just going to bounce around each other. So serine is neutral once again, and although not an amino acid, serine is almost exactly average in size. It has a molecular weight of 140.09 moles, which is only 0.3177% away from being the dead even between the smallest and largest amino acid. So a little bit of comparison between acetylcholine and serine. The mutated side chain of serine can participate in hydrophobic interactions, and it has a formal charge of 0. So even though it forms hydrogen bonds, it's not removed from the site of hydrolysis, and that's what makes it such a deadly nerve agent. This is also contributed to the fact that it's miscible in water. The original chemical, acetylcholine, can be hydrolysized to block messages between muscle and nerve cells, and it can also form many hydrogen bonds. Seeing as it is polar, this allows for it to be broken down quickly and for the body to return to normal functionality as soon as possible. So serine is a barrier, acetylcholine is a necessity, and that's the difference. This will not leave the site of hydrolysis. Acetylcholine barely needs to be there to be hydrolysized. So how does it affect the function of the cell and the organism? The protein's function is rendered useless. It cannot do its job while serine is present. The function of the cell, it will continue to produce acetylcholine as a signal because it's like, dude, no one's getting to it. It's not being broken down, so that signal is going to continue to be produced. As for the function of the organism will continue to expel nerve agents or other chemicals that are no longer present because of the nerve agent. So you'll continue to cry, to have a runny nose, etc. My protein model is limited in that it does not show tertiary structure. As you can see here, it only shows secondary structure. So there is also no globular shape. This can create a technical lack of understanding. The model is also difficult to follow to an extent because even though it's color-coded in alpha helices and beta-pleated sheets, it's not color-coded in a way that you could trace the lines. And it can be difficult at times to understand which molecule reaction is occurring because even though you know this is a methyl group, without being told that, it's hard to understand that's carbon and these are the 3 hydrogens around it.